Good morning, good afternoon, and good evening, everyone. And thank you for joining us for our webinar titled CRISPR-Enhanced Adipocyte Browning, a Cell Therapy Approach to Treat Metabolic Disease. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. This program was developed through a collaboration between the American Physiological Society and Miltenyi Biotech in support of their shared mission to provide education and value to researchers everywhere. We're very fortunate to have Dr. Michael Czech with us today, who is the Isidore and Fanny Foxman Chair of Medical Research in the Program in Molecular Medicine at the University of Massachusetts Medical School. Today, Dr. Czech will discuss a novel CRISPR-based cell therapy to induce adipose tissue browning as a treatment for metabolic disease. He will also present recent data using light sheet microscopy to visualize functional changes in adipose tissue depots. And in fact, the great image on the screen here is one of his own, so stay tuned for more of that. And with that, I'm very pleased to welcome Dr. Michael Czech. Mike, thanks so much for joining us today, and the floor is yours whenever you're ready.
Well, thanks, Liam, for the introduction, and hello, everyone. I'm delighted to be here today, and I look forward to some exciting discussions after my comments. I noticed that the American Physiological Society was formed in 1887, so many years ago. And for the last 130 years, it really has supported biology and biomedicine sciences for its members and also beyond. So it's really it's really terrific to be here today and to join this tradition. Now I'm going to talk about a topic I think that is arguably one of the major challenges of the century, and that's type 2 diabetes. And the importance of it is illustrated in part through the comorbidities on the right-hand side of the slide that are in of themselves major maladies of high seriousness. Type 2 diabetes is associated with obesity and the overweight syndrome, which makes it a metabolic disease of very high interest from a medical point of view, but also from a biological point of view. Its etiology involves two major pathways that are dysfunctional. One, the insulin signaling pathway, whereby insulin resistance occurs in the obese syndrome. And then coupled with that is a relative impairment in the beta cell's ability to secrete insulin. And the combination of those two effects leads to the glucose intolerance, the lipid metabolism abnormalities and protein metabolism abnormalities that occurs in type 2 diabetes and the comorbidities that I mentioned. Now, its connection to obesity and lipid accumulation makes it a logical approach to think about lipid synthesis, lipid sequestration as a therapeutic target. And I'm not going to go through all of the statistics of type 2 diabetes, which I think many of you know already in terms of its preponderance in the Western world and beyond. But I do want to make one point in terms of general therapeutics related to this issue of targeting fat metabolism. And that's shown on this next slide. And in a way, it's a very optimistic slide because in heart disease, in cardiovascular disease, the advent of statins, which of course, as you know, lower cholesterol and triglyceride lipids in the blood, have been associated with a really dramatic decrease in incidence of cardiovascular disease. And of course, lifestyle changes have also played a role in this. But meanwhile, in terms of diabetes, whereby only about 1% of the population had the disease in the 1940s and early 50s, that has skyrocketed to about 10% of the population in the US today. And the comorbidities that I mentioned have also skyrocketed in the same modality in the last couple of decades in particular. For example, foot ulcers and amputations for such are up to 75,000 per year in the US. So in spite of literally thousands of laboratories in the academic world and companies that have targeted this disease, there's still a long way to go in terms of prevention or a alleviation of the disease. In a way, there's optimism here because of the success in cardiovascular disease. But I think, again, we need to be using new ideas, out of the box ideas, perhaps, in terms of trying to get at the diabetes syndrome. Now, before I talk to you about the specific strategies that we've approached in our own laboratory, I want to spend some time in the introduction to make a really important point. That is the context of our strategy. And that point is that the adipose tissue, or tissues, I should say, are really a major metabolic regulator for the whole body. That is, systemic metabolic regulation depends upon adipose tissue function. And it's a major driver of dysfunction as well. And there are four, sort of bodies of work, maybe additional as well. But there are four that I'm going to hop on in terms of trying to convince you that adipose tissue is such a major regulator and is a logical target for therapeutic strategies. And the point I would make on this general theme would be that not only obesity, that is the overabundance of adipose tissue, but also the underabundance in lipodystrophies, for example, which are apparent in humans through genetic mutation, for example, or also environmental and behavioral changes, for example, in anorexia. So in the absence of adipose tissue in these lipodystrophy syndromes, you get a similar type 2 diabetes syndrome that's virtually identical to what one finds in obesity. For example, the fatty liver and the associated inflammation and fibrosis that occurs in what's called the NASH syndrome is very, very similar in the lipodystrophy syndromes as it is in the obese condition. So too little fat, too little adipose tissue, or too much adipose tissue clearly leads to the same metabolic problem and disease. So these kind of data suggest that the right amount of adipose tissue is required for metabolic health, or maybe the right types of adipose tissue and the right combinations of adipose tissue types may be the most important. Secondly, it turns out that at least in experimental mouse conditions, and these data I'm going to show you here or talk about here are from mouse experiments. I want to emphasize that. It turns out that implantation of adipose tissue can actually alleviate the disease, the type 2 diabetes that occurs in animal models, both in the case of lipodystrophy and also in the case of obesity. So on the left-hand part of the slide, it's been well documented that white adipose tissues implanted into mice that are lipodystrophic, for example, the really nice work of Mark Reitman many years ago showing this, and this alleviates the diabetes. And a major ingredient of that alleviation is the secretion of leptin. And of course, now we know that adipose tissue is really very much like an endocrine tissue in a sense of secreting many factors. Now, the alleviation due to white adipose tissue inflammation goes beyond leptin, depending on the conditions, but it is a major factor in its effect. On the right-hand side of the screen, implanting a different type of adipose tissue called brown adipose tissue has been shown to alleviate diabetes in obese mice. Now, brown adipose tissue, as many of you probably know, is a thermogenic adipose tissue. It's got high mitochondrial content. It expresses an uncoupler protein called UCP1, which uncouples the mitochondria, causing thermogenesis, causing heat, and also secretes many factors that are beneficial, that are somewhat unique to brown adipose tissue. And for those reasons, it's thought that in the obese condition, implanting brown adipose tissue, and again, many laboratories have done this. Laurie Goodyear's lab is one example of this type of work. It's been very very successful. So in these mice studies in the literature, they basically show that adipose tissue, at least in mice, can act as a therapeutic and have major effects on bringing either obese or lipodystrophic directed diabetes back to a normal metabolic profile. Now, a third point to be made along these lines is illustrated by the fact that adipocytes themselves respond to insulin. I think, as again, many of you know, the insulin receptor tyrosine kinase phosphorylates intermediate proteins, IRS proteins, and tyrosines. This activates a PI3 kinase pathway that ends up with the activation of a protein kinase AKT. And this major signaling pathway is the conduit for many or most of the metabolic sequelae of insulin signaling. And many of these relate to fat deposition within the adipocyte. So the inhibition of lipolysis, for example, which is a process which releases fatty acid from the triglyceride that's stored in the fat cell. The stimulation of fat synthesis, de novo lipogenesis, for example, and even the activation of glucose transport, which provides substrate for the de novo lipogenesis that occurs in the adipocyte. All of these metabolic pathways at the downstream region of the signaling elements are pushing the accumulation of lipid in the form of triglyceride in the adipocyte. But the main point here is shown in this next slide, relevant to the context we're talking about in terms of adipocytes being a major regulator of whole body metabolism. It turns out that this signaling of insulin in the adipocyte itself is a causative factor for systemic regulation. So, for example, if one performs adipocyte-specific knockouts, so this would be under the adiponectin promoter, for example, for the CRE, of the insulin receptor itself, or the targets of the receptor, the substrates, or the PI3 kinase itself, or AKT. The knockouts in adipocytes selectively of these components of the insulin signaling pathway, yes, they inhibit insulin signaling within the adipocyte, but they all uniformly cause a decrease in insulin sensitivity systemically as well, and they cause insulin resistance in the liver and in the muscle. Conversely, knockouts selectively in the adipocytes for negative regulators of the insulin signaling pathway. So, for example, P85 of the PI3 kinase complex, P10, which is a degradative pathway for the PIP3, or other elements downstream that are negatively affecting the sequelae of insulin signaling, they all activate the insulin signaling pathway within the adipocytes in mice. But in addition, systemic insulin sensitivity is also enhanced, and you get increased glucose tolerance in these mice. So the adipocyte itself, in terms of its signaling modality for insulin, is a causative process, presumably causing crosstalk through a variety of means, and the secretion of factors being one of them, to the liver and to the muscle and other tissues that modify glucose sensitivity systemically. Now, how does that happen? There are two major hypotheses in the field. One is the fat sequestration itself that the insulin signaling pathway affects. So the idea of sequestering triglyceride and other lipids in the fat cell, within the lipid droplets, away from the liver, away from the muscle, where they can become lipotoxic, is one modality which has been touted by many hypotheses in the literature to be a cause of increased sensitivity due to the healthy fat cell or adipocyte. Secondly, the secretion of beneficial factors, which I've mentioned, also seem to play a major role in that many, many different factors are secreted from adipocytes, and many of them are beneficial, again, to liver and muscle. And these topics have been reviewed recently in a review in molecular metabolism. Now, a fourth topic of experimental data that reinforce this idea of adipose tissue as a major driver of systemic glucose homeostasis and metabolism, is what I've alluded to earlier, and that's the brown adipose tissue depot. And that depot has been known for a good bit of time to be involved in major metabolic effects that can affect whole body metabolism. And in our laboratory, we've recently studied the brown adipose tissue in mice by a microscopic methodology whereby the actual mice can be extracted after sacrifice by organic solvents and cleared. And the methodology that we have used in our laboratory, and I want to call out Felipe Henriques and Mark Kelly for the beautiful work they've done that I'll show you, is using a method that is called the Adipoclear that was published by the Rockefeller Group, Paul Cohen's laboratory, for adipose tissue itself. With modifications that Felipe and Mark have made in our lab, one can do this with an entire mouse, in this particular case, a very young mouse. In the middle panel, you can see the fairly transparent outline of such a mouse after appropriate extraction and clearing, so to speak. And then these samples can then be analyzed by light sheet microscopy, which can then provide images that can be deconvoluted and analyzed in 3D. And so the results of this process, for example, to visualize brown adipose tissue, as we have used it for, results in a image of this type, where you can see upon sort of marinating the tissue with anti-UCP1, which is selective for brown adipose tissue, a very, very nice imaging of the brown adipose depot in the neck region, in the interscapular region of this animal. So these data and this approach has been very valuable to not only locate brown adipose tissue, but also in follow-up studies, which I'm not going to be able to show you, which are in progress, to outline other cell types that are involved in the brown adipose tissue depot. And this is a magnification of the same animal sample that I just showed you. So the brown adipose tissue coordinates with white adipose tissue, at least in mice, in terms of driving whole body metabolism. I've already mentioned fat sequestration, which is a feature of the white adipose tissue, the secretion of adipokines from the white adipose tissue. But brown adipose tissue also has a profile of secreted proteins and other factors called the badokines. And both the badokines and the adipokines can modulate liver metabolism by, for example, inhibiting the lipid synthesis in the liver, and also the release of glucose, and also the uptake of glucose in the muscle. So in addition to fat accumulation or sequestration and adipokine activity, the thermogenesis of brown adipose tissue also potentially can contribute to the utilization of fatty acid and glucose itself in maintaining metabolic homeostasis. Now, in addition to the brown adipose tissue, again, as many of you are probably familiar, brown adipocytes, sometimes called beige adipocytes, can also occur in the white adipose depots, especially upon cold exposure of mice or humans for that matter. And the beige adipocytes, which occur in these white adipose tissue depots, particularly the subcutaneous white adipose depots, also have the ability to express UCP1, the uncoupling protein, produce heat in a thermogenic pathway in response to cyclic AMP, and therefore enhance energy expenditure, but also secrete unique beneficial factors that also affect liver and muscle in terms of modifying whole body metabolism. Now, the white adipose depots that contain beige adipocytes and themselves quite complicated, containing other cell types, immune cells, for example, and are highly innervated by the sympathetic nervous system. And it turns out that the sympathetic innervation of white adipose depots are critically important. Matter of fact, absolutely required for the appearance of beige adipocytes, for example, in cold exposure. And so, again, our laboratory has been interested in studying these interactions of the innervation and the other cell types within adipose tissue. And again, we've turned to light sheet microscopy to analyze those interactions. You can actually see the beige adipocytes, or at least the Beijing of the adipose depots in the cold, by visual inspection of the subcutaneous adipose depots within a mouse, for example, that's been incubated, that's been housed at room temperature versus cold exposed, as you show here. This inguinal white adipose tissue depot, as you can see in the middle panel, is contracted. It looks brown. It's more vascularized, even to the naked eye. And importantly, if one denervates that depot, again shown in the right panel, with a chemical 6-OTA injection specifically within this adipose depot, one can prevent virtually completely the upregulation of UCP1 and the visual beige look of the adipose tissue. And this is shown in the bottom panels, where the histology slides have been stained with UCP1 antibody. So the middle panel shows a region of Beijing, for example, in the white adipose tissue, which is completely blocked upon denervation. So to study this in more detail, as I mentioned, we've gone to light sheet microscopy, again, using the methods of the Rockefeller group, Paul Cohen's group. And I should mention that his group has also published very nice data along these lines, as well as Wenwen Zhang laboratory has also published data along these lines if anyone wants to take a look at the details. Upon clearing the adipose tissue, and this is about a 1 or 2 centimeter length inguinal adipose tissue depot from mouse, you can see by using the anti-tyrosine hydroxylase antibody to stain the sympathetic nerve fibers within the adipose tissue, a very strong network and detailed network of sympathetic nerve fibers within this tissue. And as I will show you in a moment, these nerve fibers really are extensive enough to be in proximity with virtually every adipocyte, at least a beige adipocyte within the process. Now, beiging the IWAT, inguinal white adipose tissue in the mouse by cold exposure or other means allows one to visualize the browning effect by using double antibodies. So the TH antibody to visualize the innervation, and anti-UCP1 antibody to visualize the brown adipose tissue or brown adipocytes or beige adipocytes within the tissue. And you can see upon beiging, there's a extensive production of cells or appearance of cells that are expressing UCP1 under these conditions, which again is very nicely illustrated and in these light sheet microscopy images. Now, in our laboratory, we've been able to go one step further and use a 12x objective to increase the magnification resolution. And again, I want to shout out Felipe Henriques for the wonderful work he's done in our lab in terms of this light sheet microscopy and imaging. So this is a segment of the subcutaneous white adipose tissue in a mouse, a segment of what I showed you previously. And it shows, again, the really intense network of sympathetic nerve fibers and the individual brown or beige adipocytes that you can see now that become resolved at this level of magnification. And as the image zooms in here, you'll be able to see the individual, sort of in the middle to the right, individual adipocytes with the bulges coming out, indicating that the lipid droplets within the beige adipocytes are pushing out at the surface membrane. So these kinds of analyses have really allowed us to begin to really analyze the relationship between the innervation and the brown adipocytes. And as I mentioned, the innervation is absolutely critical. A single optical plane also shows this, where the beige adipocytes are juxtaposed to the nerve fibers, sympathetic nerve fibers, that virtually are next to virtually every beige adipocyte that one can visualize in these images. Now, these kinds of data have been further improved or further extended by a number of laboratories, including our own. And we've recently reviewed these type of data. But I want to point out two other features of this system. One is the idea that not only is there innervation that's very extensive in the adipose tissue, but also the adipocytes themselves can speak to and enhance the innervation. And that has been shown recently by a paper from the Paul Cohen Laboratory, cited here on the left-hand side of the slide, which shows very nicely such an effect. In addition, the adipocytes secrete a good number of factors, shown on the right-hand side, which are known to affect, at least potentially, sensory nerves, which also innervate the adipose tissue and therefore can carry signals back to the brain and modulate the central nervous system under these conditions. So this effect allows, we think, adipose tissue depots to actually crosstalk and speak to other depots distantly. So the Tim Bartness Laboratory, through many papers, as well as other laboratories, for example, have shown that lipolysis by a white fat depot, a subcutaneous depot in the mouse, can actually lead to the triggering of sensory neurons, which then activate distant brown adipose tissue depots to become more sympathetically activated and more active in their heat production. Recent work in our laboratory, spearheaded by Adilson Gulhermi, have also confirmed that kind of effect, but have also been able to use a method for inducing the sensory neuron stimulations by knockouts within the adipocytes themselves. And again, this paper by Adilson, who led the work, it can be seen in Molecular Metabolism for more details. Now, one last superimposition on this overall network of modulation is a nice study by Jiandi Lin's laboratory at University of Michigan, for example, which shows that upon activation of the brown adipose tissue by these kinds of long-term mechanisms or other mechanisms, can be used to secrete factors such as NRG4, for example, which are known to be able to modulate the liver, in this case of the Jiandi Lin publication, to decrease de novo lipogenesis and enhance glucose tolerance by virtue of NRG4 secretion. So you have a secretion modality superimposed on a neuroendocrine modality for adipose tissue to potentially modulate important metabolic tissues and maintain systemic health. Okay, so everything I've said so far is mostly involved work on the mouse, but it also refers, I think, directly to the human condition, because it's been almost now 20 years since the original findings from PET scanning and imaging showed that beige or brown adipocytes are also present in the human anatomy, and particularly around in the neck region. This is variable from one person to another. Some have more, some have less. But one point that's been made over the years by, again, a number of laboratories is that the amount of apparent brown or beige adipocytes and the abundance of such adipose correlate positively with metabolic health in humans. And I should have pointed out in the mouse studies that I showed, this has been demonstrated more directly by knockouts, for example, in Bruce Spiegelman's laboratory, that nicely showed the efficacy of beige adipocytes in the mouse to actually directly affect insulin sensitivity and glucose tolerance. This is a correlation in the human, but also a very tight correlation. Importantly, very recent data by the Cypress lab at the NIH has shown more of a direct causation by activating the beta-3 adrenergic system in humans with agonist and showing that not only can that enhance a Beijing effect, as sort of showed by these images here, of the beige adipose tissue, but also activate insulin sensitivity and glucose tolerance in human subjects along these lines. So this is a very exciting set of findings, not really at the therapeutic stage at this point. There are side effects that are observed in terms of overlap to other tissues in terms of the adrenergic stimulation, apparently. But I think this is an area that is being continued to be developed and has interesting potential in terms of therapeutic strategy. But I want to highlight in particular this last point, number 3, at the bottom of your slide here, and that's the idea that Silvia Corvera's laboratory at the University of Massachusetts has promoted with some really beautiful data in a paper a few years ago in Nature Medicine, and that's the results of implanting human beige adipocytes in immunocompromised mice, which therefore don't reject the human adipocytes, showing that it improves glucose metabolism to do so. And that's more detailed, outlined in terms of the experiments that her laboratory has achieved in these publications that, again, you can take a look at in more detail. The basic finding of these papers that's most important to the discussion today is the ability to greatly expand the adipocyte progenitor cells within relatively small samples of adipose tissue, perhaps up to 1,000-fold expansion. And this expansion of progenitor cells then allows, of course, the cells to be differentiated in vitro into beige adipocytes, or a combination of beige adipocytes and white adipocytes, and then to be implanted into immunocompromised mice. And as I've mentioned, showing that improved glucose homeostasis can be achieved by this. So these data suggest strongly a therapeutic strategy that may be analogous to the CAR T cell therapy modalities that have been so successful in cancer biology, whereby the idea being that ex vivo manipulation, in this case expansion of adipocyte progenitors, could then be used to produce more therapeutic adipocytes and enhance the number, the abundance, when perhaps implanted back into the same patient. And into the same patient is important because, again, there would not be an immune barrier for such implantation and engraftment. So this idea is an idea that we have used to extend in the experiments I'm going to tell you about today, with a additional step, and that is a set of experiments and technology we develop in our own laboratory, whereby we can take progenitor cells, adipocyte progenitor cells, and use CRISPR technology to enhance their Beijing even further. And I think, as many of you are well aware, CRISPR technology involves a protein like Cas9 bound to a guide RNA. And when such a complex is formed, that guide RNA can target the complex to a gene or a region of your choice based on the sequence of the guide RNA. That guiding of the complex allows the Cas9 protein to cause or mediate a cleavage of the DNA, and then the DNA repair mechanism fosters the insertions and deletion mutations that occur during the repair. And these insertions or deletions, called indels, can then disrupt the reading frame of the gene of choice and even form a stop codon to prevent the full synthesis of the disrupted protein. So these kinds of approaches, and there are other kinds of modulations that, of course, the CRISPR and Cas9 and other Cases can modulate, but this is the simplest form of CRISPR technology that we've used that I'm going to describe for you. So taking these two technologies together, the technology in the Corvera lab of being able to use small adipose samples and through using angiogenic conditions greatly expand the generation of progenitors, and marrying that technology to the technology we've developed in our laboratory on fine-tuning and increasing the efficiency of CRISPR-mediated gene disruption, we have targeted the goal of using these two methods to then make or produce large numbers of enhanced beige adipocytes, enhanced by the CRISPR technology for implantation to determine whether or not we can improve metabolic health in the recipient mice. Now, to be able to affect a therapeutic CRISPR method requires the execution of a number of objectives shown on this slide. First of all, we would want to identify adipose suppressor genes so that when the suppressor genes are knocked out, you have increased browning or beiging of the adipocytes. You want to use a CRISPR technology that must be stealth. That is, after affecting the gene disruption, you want the disappearance of the Cas9 protein and sgRNA so that you don't have immune reactions against the Cas9 protein after implantation. We want to reduce off-target effects that might compromise the browning effect. And most importantly, one wants to develop a methodology for pre-adipocytes or adipocytes that is very, very high efficiency ex vivo. In other words, that virtually 100% of the cells in an ex vivo cell culture would be targeted and affected in terms of their increased beiging. So we've developed methodologies to satisfy these criteria. And the first point I want to talk about, of course, is identification of the browning suppressor gene that we have targeted. And that gene is shown here, and we found it. It's called an NRIP1 or RIP140 as the protein is denoted. It was discovered in our laboratory many years ago, 2006. We had our first publication in an RNAi screen where we were screening for genes that, when they were knocked out, enhanced insulin signaling in the adipocytes in culture. And we found this hit, as you can see here in the middle of this relatively small screen. We actually did 2,000 or 3,000 genes over a course of several weeks in this screen. This is one of the largest hits that we had, or biggest hits that we had in terms of upregulation of insulin sensitivity. And it was a gene which not only increased insulin sensitivity, but was then found to enhance the overall metabolism of adipocytes. And I should say that the Parker group in the UK also discovered this gene and published a paper on knockout of RIP140 in 2004 in PNAS, and then followed this up with a number of experiments and review articles, as we have as well in this paper that I'm citing, the Pawelka et al. JCI paper. The bottom line is RIP140 turned out to be a suppressor of nuclear receptors, which activated glucose utilization, mitochondrial functions, including the upregulation of the uncoupler protein UCP1, which I've described, including the activation of fatty acid esterification and oxidation, fatty acid oxidation being a prime target, and also the secretion of beneficial factors. All of these genes in many, many different pathways are upregulated upon knockdown of RIP140. So this is a ideal gene to target in a scenario that I've described for you, and I will focus all the attention in the remaining slides on RIP140 as the target gene that we've chosen to study. The first series of experiments I'm going to describe in this context will be actual mouse-to-mouse experiments, because we wanted to develop the methodology first in the mouse system and then apply it to human adipocytes. So in these kinds of studies, we've taken adipose tissue from 2 to 3 week old mouse pups, digested out the cells, adipocytes from non-adipocytes, the non-adipocytes, so-called SVF fraction, is then centrifuged, plated. That contains the progenitor cells. And then we've used a Cas9 sgRNA complex methodology for targeting these cells and knocking out NRIP1. And I'll show you the methodology in a moment. And then we have differentiated these cells, and the NRIP1 thankfully has no effect on the differentiation, does not inhibit differentiation prior to implantation into mice. Now, the first step we made in this protocol was, of course, to identify sites within the NRIP1 gene that would be applicable in terms of disruption by the sgRNAs. And NRIP1 is quite a, if not unique, very distinctive gene in that its entire reading frame, as you can see in the upper right-hand corner, is encoded within exon 4. And we targeted multiple regions, as you can see in the lower part of the slide, where we have the mouse and the human sequence shown, and the sites of the sgRNAs for both the mouse in green and the human in red. And we identified several sites upstream, but also downstream in the gene that were amenable for targeting based on availability of PAM sequence and other aspects of the overall sequence. And in using one of these sgRNAs in complex with the protein Cas9, we settled on electroporation as a method for transfection. Many of these other methods that are available for executing CRISPR technologies ex vivo depend on the continued expression of Cas9, and that's not an acceptable modality under the conditions that we are studying. So electroporation of Cas9 protein and sgRNA, so RNPs, is a very acceptable methodology, because as you can see in the Western blot shown in the middle of the slide here, the Cas9 that is introduced within the cells by the electroporation over the next 2 or 3 days pretty much is degraded and disappears, and by about 3 days out, there's no detectable Cas9 protein. And then by 11 days, at the very last right-hand lane, after differentiation, you can see a very nice upregulation of UCP1. So these kind of data then satisfied the criteria of very nice gene disruption of the RIP140, but the absence of Cas9 after the differentiation process. On the right-hand bar graph, you can see the very high efficiency. Once we worked out the conditions for the voltage of the electroporation, and also for the ratio of Cas9 versus the sgRNA, we can get 90 to 100% efficiency, 90 to 100% of the cells in a culture disrupted, as evidenced by the Sanger sequencing that we can do around the predicted regions of indel formation under these conditions. But we then found a very interesting result, which was initially unexplained, which is shown here. And that result stems from the analysis of all the different sgRNA complexes that we made, as shown at the very top diagram here. And each of these sgRNAs that were combined with the Cas9, as shown in the upper left-hand bar graph, all showed very, very high efficiency of gene disruption, of NRIP1 disruption, in the locations at which they were targeted, again by Sanger sequencing. But interestingly, as you can see in the lower pieces of data, on the left lower of the RT-PCR, the UCP1 upregulation, which is quite dramatic for many of the sgRNAs, were absolutely absent. There was no UCP1 upregulation for several of the sgRNAs, especially in the upstream regions of the gene. And this was corroborated by the Western blot you can see in the lower right-hand region, where the UCP1 upregulation is seen for all of those sgRNAs, sort of from the middle to the downstream areas, whereas the upstream sgRNAs had no effect on upregulation of UCP1. We were very confused by this because of the high efficiency of all the sgRNAs in disruption. But the presence of the ineffective sgRNAs in terms of upregulation of UCP1 all occurred very upstream. So these data suggested the possibility that there were initiation sites downstream from those that were actually still producing some RIP140. And this was corroborated by IP of RIP140 in these different samples. And as you can see, those samples which contain the sgRNAs where there was no upregulation of UCP1 show the residual RIP140 protein that are in those samples, whereas the samples where we have very nice upregulation of UCP1 show the complete absence of RIP140 protein. So these data suggest that for those of you who might be doing similar experiments, one might have to use a number of different locations within the gene or in different exons to get a full knockdown of your protein if you're using it in this modality. Now, what happens when we tested these adipocytes that were knocked down for NRIP1 compared to controlled adipocytes when implanted into mice? And that's shown on this slide. And these experiments we're implanting either controlled adipocytes or NRIP1 knockout adipocytes. And these experiments, I should say, were spearheaded in our laboratory by Emanuela Sakaraki and also Sarah Nicolaro in the laboratory, in collaboration with a number of other labs, which I'll cite in a few moments. Now, the implantation of these mice were then continued on chow diet and then implemented with a high-fat diet several weeks later, and then let to move forward on a high-fat diet for several additional weeks, as shown in this protocol here. And when we take a look at the implants after such a protocol, we can see that there are 1 to 2 centimeter long adipose depots that form from these adipocyte implants, very much confirming the initial data from Silvia Corvera's laboratory in that Nature Medicine paper that I had cited, showing very, very nice adipose depots emanating from such implantations. Now, most happily for these experiments, when we analyze the NRIP1 implants for UCP1 expression, as you can see in the lower left-hand panel, we see a resilient upregulation of UCP1 that is retained throughout all the weeks of implantation under these conditions, showing that the NRIP1 knockout has indeed caused beiging of the cells, which can be retained within a long-term implantation protocol such as this. Now, interestingly, I'm not going to show you the data, but we've also done RNA-seq and other analyses for gene profiles, gene expression profiles in these implants. And not only UCP1 is upregulated, but a huge set of Beige genes or Beige associated genes are also upregulated under these conditions. And I won't show you all that data. So this beiging is a very global beiging effect of the NRIP1 knockout, consistent with older data that we and others have published over the years on this system. Now, we can also analyze, again by light sheet microscopy, these implants to ask the question, how many of the cells that have been implanted are actually expressing the UCP1? Is it uniform throughout all cells, or there's only certain cells that are expressing the UCP1? And that light sheet microscopy is shown in this image here of an implant similar to what I've just shown you, a section of the implant. And you can see in this staining for UCP1 that a good number of cells, dozens of cells in the implant, are highly expressing the UCP1 and those other genes, presumably, that I mentioned that are associated with the beiging phenomenon under these conditions. So again, the maintenance is quite dramatic. And of course, one of the major goals in our collaboration with the Corvera lab is to now enhance the beiging even further beyond this, so that even a higher abundance of beige cells present in the implants and in the in vitro system, and then in the implants after putting them in vivo. We're not able to see innervation in the original implant that I just showed you, based on the autofluorescence of the matrigel that's used. But in subsequent experiments, as shown here, with a higher magnification, we do see some evidence of innervation of these implants, and also the vascularization that the Covera group showed in the original experiments is also visible under these conditions. So taken together, we think that these data show a very nice effectiveness of implantation of adipocytes under these conditions. Now, what about actual function? Well, analyzing these animals again showed a encouraging result. For example, as shown in the right-hand upper panel here, you can see that the black bars indicating adipocytes which had been treated with Cas9 sgRNA, but with a non-targeting sgRNA, or untreated mice shown here, show an increase in their blood glucose upon the high-fat diet exposure, whereas the animals that have the implanted NRIP1 knockout adipocytes do not show that increase. So apparent increase in glucose tolerance shown here. And this is corroborated by small but highly significant enhancement of glucose tolerance in glucose tolerance tests, GTTs, and also in ITTs, insulin tolerance tests, on the right-hand lower panel under these conditions. Now, probably the most dramatic effect we've seen, in addition to the enhancement of glucose tolerance and the lowering of fasting blood glucose levels, is the decrease in fat accumulation in the liver. And as you can see, the animals at the top left panel, which have control adipose tissue implanted, are quite laden with fat, whereas the animals with the NRIP1 knockout adipocytes implanted are much less visually fat laden. And this is corroborated by the middle panels of histology, showing very large lipid droplets in the control animals, whereas in the NRIP1 knockout, you can see a dramatic decrease in triglyceride, again corroborated on the very right by triglyceride analysis biochemically. Now, in collaboration with the Covera group, we have further extended this with the human adipocytes. And again, we've used sgRNAs targeting similar locations in the human NRIP1 gene, as shown on the top left cartoon. And again, right below that, you can see in the bar graph, most of the sgRNAs under these conditions were seen to be very high efficiency. And these experiments, again, driven by Manuela and Sarah, showed a very similar pattern of upregulation of UCP1 in the sgRNAs that are similar to the ones we found that were able to upregulate UCP1 in the mouse. So the same phenomenology occurs in the human, where sgRNAs up in the upstream region of the gene do not show an increase in UCP1. And we're just now analyzing the RIP140 protein under these conditions. And our expectation would be, again, similar to the mouse condition. We used a protocol for these experiments whereby the human adipocytes that were obtained through expanding the progenitors from the human adipose samples, and then modified by the Cas9 process that I've just described to obtain beige adipocytes, and then implantation into immunocompromised mice in a protocol shown on the right-hand side was utilized. And these mice are NSG mice that are held at thermoneutrality to enhance their glucose intolerance under these conditions. And as you can see in the bottom left, the experiment with human adipocyte actually shows a small decrease in body weight in the NRIP1 implants, and also in the middle panels, a significant enhancement of glucose tolerance using GTT analysis, shown in the bar graph on the right, being highly significant in the improvement in glucose tolerance. And then finally, in this series of experiments, we can show a decrease in liver to body weight ratio, and also a decrease in liver triglyceride under these conditions. So the protocol that I've described, using human pre-adipocytes, progenitors as a starting material, and then the implantation along the lines of comparing the NRIP1 versus the control adipocytes, really show very similar effects as we've seen in the mouse-to-mouse experiments. So to summarize these, what we think are very encouraging results, the CRISPR RNPs can edit human and mouse primary adipocytes and pre-adipocytes with high efficiency. We are able to show that deletion of the co-repressor NRIP1 induces adipocyte UCP1 and the full Browning profile with examples of many other genes that are upregulated that are associated with the Beijing effect. Implanting CRISPR-enhanced NRIP1 knockout adipocytes forms very nice adipose depots, and these depots, upon their formation over the long term, over weeks of high-fat diet, prevent the high-fat diet-induced glucose intolerance and hepatic steatosis that one sees when in animals that are untreated or animals that are implanted with normal adipose cells. So these data to us are very encouraging, and we're now trying to extend the proof of concept that I've shown you here with mouse experiments, moving towards non-human primate experiments. And we're excited about continuing the collaboration with the Corvera lab and other laboratories in pursuing those future goals. So I'll stop here and thank the many colleagues and collaborating labs that have been involved in these experiments. I've tried to outline the individuals in the laboratory that have spearheaded the experiments that I've shown you, but I have to say that our entire laboratory have participated in one way or another in great discussions and in helping out with many of the aspects of the experiments that I've described today. We also want to, of course, highlight our close collaboration with the Corvera lab and the members of her lab that have also tightly collaborated their efforts and our own. The Eric Sonheimer lab, Scott Wolf lab, who produced the Cas9 protein and other help for the work. The Jason Kim lab, Randall Friedlein, and also the Paul Cohen lab at Rockefeller that have helped us so much, and Jingyi Qi in particular helped us so much in developing our light sheet microscopy capabilities now that we are pursuing in our own laboratory at UMass Medical School. So with that, I will stop, and if there are any questions, we'll be happy to have further discussion.
Certainly, yeah. Thanks so much, Dr. Czech, for the really fantastic presentation. But let's get started here. First question, Dr. Czech, you showed that Beijing of white adipose tissue is blocked by denervation, but your lab has also shown that immune cells can play a role. So how is this coordinated?
Yeah, excellent question. Indeed, we have shown that a Beijing phenomenon that can be induced by a knockout of fatty acid synthase in this particular case, as well as other labs have shown this, can induce a Beijing. And in our lab, we've been able to show that this occurs even in denervated adipose tissue. We've tracked this down, again, work by a number of people in the lab, spearheaded by Felipe Henriques, has shown that indeed immune cells are involved, and the immune cells are likely collaborating with adipocytes, but other cell types as well. So this is a complicated situation, and we're in the throes of trying to identify factors that communicate between the adipocytes and the immune cells, and in particular, factors that are secreted by immune cells and perhaps can directly modulate UCP1 regulation. But I think, in summary, I think it's important to recognize that under physiological conditions in terms of cold exposure, the innervation by the sympathetic nerves in particular are absolutely required for the beiging. So that physiologically, if the immune cells are involved, and they do seem to be, many labs again have contributed to this, they probably are interacting with or in addition to the sympathetic nervous system.
Fantastic, great answer. Next question. As far as adipose beiging or increasing brown adipose tissue in human is concerned, is it your understanding that chronic mild cold exposure or short, frequent, more extreme exposures are more effective?
Well, at least in mouse experiments, the cold exposure as short as a day or so already shows the beginnings of the beiging process. But this continues to get more and more extreme over, for example, a week or even 2 weeks of cold exposure. So the longer term cold exposure does give you more extensive beiging and activation of the thermogenic process.
Excellent. Would overexpression of adiponectin cause the same metabolic effect as increasing white adipose tissue browning?
We don't feel that adiponectin is a strong cause of beiging. There may be effects that it exerts, but again, compared to cold exposure, I'm not aware of effects of adiponectin that would be as strong an inducement of beiging than the normal cold-induced sympathetic nervous system activation.
Perfect. Next question here. Is the effect of NRIP1 knockout in adipocytes additive to cyclic AMP signaling to upregulate UCP1?
Yeah, excellent question. Manuela in the lab has worked on this system as others have as well. And the bottom line is that the NRIP1 pathway, which regulates so many different genes, is a very strong browning effect, but it's pretty much independent of the effect of the cyclic AMP signaling system. So cyclic AMP signaling also induces browning by independent pathways. And one of the most exciting findings that we have is that the two together are quite synergistic in inducing browning and beiging. And so we think there's a lot of potential for us to combine the activities of these two pathways in our future experiments to enhance the therapeutic effect.
Fantastic. Next question here. Say, hi, Mike. Thanks for a great talk. Do you find any depots that are innervated by sympathetic neurons but do not see a significant increase in beige adipocytes upon cold exposure or Melia that induces beiging? And are omental or visceral fat depots poorly innervated by sympathetic neurons? And would that explain differences between visceral and subcutaneous beiging?
Yeah, very good point. I perhaps should have even mentioned. Yes, the visceral adipose tissue, so the gonadal adipose tissue, for example, in a mouse, shows much less beiging than the subcutaneous adipose tissue under these conditions. And that may have to do with the sympathetic nervous system and the innervation. But I think it's very complicated because there are so many other factors that are involved in the beiging process. So I think this is a question that is a really important one for future studies, and we need to be looking more carefully at the details of the sympathetic innervations and the coordination with other cell types.
Fantastic. Here's a great question here. Does it matter where in the mouse the implantations are placed to attain the optimal effect?
Yeah, it's again a good question. This hasn't been fully addressed. There's more work to be done. I think Silvia Corvera's laboratory has done some really nice work related to this, but I think there's still a lot more to be learned. I think what we can say is that at least so far, our implantations in our own laboratory, in terms of the mouse to mouse, have been done mostly near the interscapular area, thinking that maybe the high vascularization in that area and maybe innervation as well might improve the implants. but we haven't noticed any differences if we have slightly different locations. But again, it's a good question. I think it needs further work to determine whether or not there may be areas that are particularly prone to high efficiency in terms of the Beijing process.
Fantastic. Yeah, great answer. Interesting question here. Could you overexpress UCP1 using CRISPR and Cas9? And do you know if this could improve Beijing?
Yeah, very good question. I should have mentioned this. There are so many labs that have contributed to this area. The Tseng lab at Harvard has published a very nice paper showing that upregulation of UCP1 alone is also effective in enhancing glucose homeostasis in mice when implanted back into mice. So this is a very exciting finding, indicating that UCP1 itself has a number of effects, perhaps including the secretion of factors, which were also discovered in that paper by the Tseng lab. And so I think this is an interesting point to be made. I would also, though, mention that the knockout of NRIP1, of course, upregulates UCP1, but many other genes as well. So we feel that the NRIP1 is particularly valuable in terms of a target for long-term therapeutics. And of course, we have a long way to go in terms of being able to determine whether this approach would ultimately be able to be brought all the way into clinical trials or not. But I think the point made here by that question is a good one, and UCP1 by itself seems to be able to initiate a number of events, including the secretion of factors that are very valuable here. So good point.
Yeah, really interesting. Here's another good question here. Did you keep any of your NRIP knockout implanted mice alive for longer than 16 weeks? And do you know if there's any concern of these brown adipocytes depleting or at least significantly reducing the energy reservoir capacity on a whole body scale if kept there for an extended period of time? Or would these cells regulate themselves over time?
Right. Yeah, good question. We haven't gone too much longer than the several months that I showed in these slides because we've sacrificed the animals for analysis, but we are doing that currently. But we haven't noticed any deleterious effects of the implants over those several months. And I think we need to do longer term experiments to fully be able to answer that excellent question. We're hopeful, of course, that the implants in mice would go out a year or even more and be effective. But the analysis of the implant that I did show by light sheet microscopy does show that there are numerous cells that are highly expressing the UCP1 over those several months, suggesting that long-term survival of these implants in the beige state seem possible.
Excellent. Okay, next question. Are there other thermogenic suppressor genes that might serve as even better targets to enhance Beijing of white adipose tissue?
Yeah, good point. There are several suppressor genes that have been published over the years, perhaps a dozen or two, that either through modulating differentiation or modulating other pathways seem to enhance UCP1 expression when they're knocked out. And we're currently involved in looking at combinations of knockouts of these other genes. We haven't so far been able to really extend the Beijing very dramatically by these knockouts. But as I mentioned, we were able to extend the Beijing very dramatically by activating cyclic AMP pathway as well. So we are now focusing on ways we might be able to enhance the cyclic AMP pathway by gene knockouts. And the obvious ones would be PDE, for example, phosphodiesterase. We're looking at that as well as other genes in the cyclic AMP pathway that suppress cyclic AMP or PKA activity, protein kinase A activity. So I think there's plenty of potential for developing further lines of investigation by multiple knockouts, and we're hoping to really do that over the next year.
Yeah, it definitely sounds like a really exciting avenue of research there. Next question here. Amazing work. How many cells are transplanted, and what are the effects, if any, or what are the effects, if any, that come from the thyroid hormone?
Interesting question. Okay, good point in terms of the number of cells that are implanted, because this really, relates to, is this ever possible in terms of a human therapy? It turns out that in the experiments that I've described, for the most part, there's several million cells implanted, maybe up to 10 million cells implanted per implant. And in the mouse experiments, there are 2 implants that we do actually nearby each other. And it's in that range, roughly in that range. In humans, the amount of beige or brown fat has been estimated. It's not impossible to estimate it exactly, obviously, but some of the estimates are around 50 grams of beige fats. That's about 0.1% of the weight of an individual. So fairly small amount of beige adipocytes or beige fat is present. And if it is therapeutic, means that perhaps a relatively doable amount of implantation of fully beiged adipocytes could be very effective, even in larger animals or humans. So it's a really interesting question. We need to do more work in terms of how much of an implant is required to give maximal metabolic effects. And again, those experiments are underway and really important.
Next question here. Do you think doing an ex vivo UCP1 induction using drugs rather than just CRISPR would have similar benefits once reintroduced to mouse or humans, including lower systemic toxicity if a drug is given orally?
Yeah, interesting idea. I think what the idea is that one would treat with a pharmaceutical the ex vivo adipose tissue prior to implantation. I think that's what the implication of the question was. Of course, if that were the case, it would have to be a treatment that were permanent. And so the upregulation of UCP1 and all the other beige associated genes would be maintained. Is this a therapeutic possibility in terms of oral delivery of a pharmaceutical that could maintain beige? That would be a terrific approach, and is a terrific approach. It is being worked on currently, but I think there are other tissues that may be responding either on target or off target to beta-3 agonists and so forth. So there's a lot more work to be done, I think, in that realm prior to really being able to move into a therapeutic mode.
Okay, here's one. Do you consider that all drugs or interventions that lead to UCP1 upregulation could be acting through NRIP1 or be requiring this suppressor?
Yeah, I don't think so. I think that there are, many roads to UCP1, so to speak. I already mentioned that the NRIP1 pathway is very distinct from the cyclic AMP pathway. I think they're probably, we know there are other pathways that regulate UCP1 as well. So I think that the excitement here is that because there are so many ways of upregulating UCP1, and by the way, it's not only UCP1, it's a number of other genes that are beneficial, NRG4, for example, and many others, that I think that the potential here for multiple targeting, I think, is particularly encouraging under these conditions. And really our lab is devoted. I think if there's one approach or one question that our lab is devoted to among the many others is the question, can we target multiple genes to bring a in vitro system of adipocytes that are mixed all the way to a full brown phenotype? That would be the goal, so that we can optimize the beiging and the browning prior to implantation.
Perfect. Okay, interesting question here. Do you know if there's any, does cold exposure have any effect on NRIP1 suppression? Do you know if there's a connection there?
Yeah, good question. There are some effects on NRIP1, but not so dramatic as one might think that the effects are being mediated through NRIP1. And I would remind everyone and ourselves that the NRIP1, of course, expression level isn't the only modality whereby NRIP1 can modify gene expression. There are many other covalent and even non-covalent mechanisms that can modify NRIP1, RIP140 activity. So I think we have to be careful of equating simply the expression levels with the activity. But I think, again, it raises a really exciting question, and that is, what are all the modulations on RIP140 that modulate its activity? And I guess, this is, again, I think based on the data we're showing here, it's a really important question, I think, for the field to address.
Yeah, certainly. And I think in the interest of time, we'll just try to squeeze in a few last questions here. What is the role of adipocyte differentiation with regards to obesity and type 2 diabetes?
Could you ask that question again?
Yes. Where is it here? Yeah. What is the role of adipocyte differentiation with regards to obesity and type 2 diabetes?
Okay, differentiation. Yeah, I wanted to make sure yeah, I think this is a really, really excellent question because the progenitors physiologically, of course, are going to be critically important in terms of the numbers of adipocytes that ultimately are expressed, the types of adipocytes that are expressed, in which depots they're expressed. So I think this issue of adipocyte differentiation and the amazing heterogeneity of differentiation and heterogeneity of adipocyte types, I should say, is really an area that's really, really fertile and very, very active right now. And I think the degrees to which different types of progenitors are differentiated under which physiological conditions and in which locations are going to be a key factor in metabolic health. So this is a great, great question and a great area.
Fantastic. We talked a bit about where grafts could be implanted, but maybe you just could clarify, where are the adipocyte grafts being implanted, and is the region innervated and vascularized, and do the grafts enhance innervation and vascularization?
Right. Again, the work that we have done is mostly in the interscapular or below the interscapular area, and where we hope that the innervation and vascularization may be, if not maximal, at least optimized. We don't have a clear analysis of the vascularization or innervation in our laboratory. I think those kind of data are being developed very strongly in other labs, and in particular in the Corvera laboratory. I think there's very exciting data coming out of that laboratory along these lines. But I think we have evidence, surely, that there is some innervation and certainly some vascularization, and probably that's really required for optimizing the overall survival of the implant. So again, it's an opportunity, I think, for raising the issue of how can we find genes that optimize those aspects of adipose tissue survival and development into an adipose tissue depot under these conditions. I think that's an exciting area, again, underdeveloped, and I think it's going to be really fun to look further into this point.
Yeah, definitely. And I think, we'll make this next question the last one. But how does Beijing on a certain fat depot, for example, in the vicinity of the pancreas or liver or kidney, how might that influence the function of nearby organs?
Yeah, good question. We don't really have information ourselves on that question. It is true that adipocytes are being found in the strangest corners, including in the pancreas, so that the juxtaposition of adipocytes to other cell types and other organs, I think, is a really important question. But unfortunately, we ourselves don't really have direct information on that important question.
Perfect. Well, thanks so much, Dr. Czech, for the really fantastic presentation and also all the great insights in the Q&A today. It's been a real pleasure having you with us.
Thank you very much, and thanks for all the great questions. Really enjoyed that.
Yeah, definitely. Big thanks to everybody for all the fantastic questions and for participating in the webinar today. Of course, we also want to give a big thank you to Miltenyi Biotech for sponsoring this event to make it all possible. And so in closing, thanks again for taking part in this American Physiological Society and Inside Scientific webinar, and we look forward to seeing you again soon. Have a great day, everyone.